What if one tiny spelling mistake in your DNA could ruin your entire life? And what if science could just edit it out? Hi, I'm Priyanka and welcome to the Science Nest. In our previous video, we discussed about the top 10 breakthroughs in the future of biology, and in this video, we'll explore gene editing and precision genomics, a technology so powerful that it changes how we treat diseases, food, and life itself. We edit everything today, starting from WhatsApp messages, Instagram captions, even our selfies. But what if we could edit genes? What is gene editing then? Gene editing means changing DNA at a specific location. Think of DNA as a massive instruction manual for your body. One spelling mistake and suddenly there is a disease. Just like one typo in your exam answer can make you fail your exams. So here is the solution. CRISPR-Cas9 or clustered regularly interspaced short palindromic repeats and CRISPR-associated protein 9, a revolutionary gene editing tool that functions like molecular scissors. Adapted from a bacterial defense system, it uses two key parts, the Cas9 enzyme, the cutting scissors, and a guide RNA that acts as a GPS directing Cas9 to a precise DNA sequence. Once the gRNA binds, Cas9 makes a clean double-strand cut. The cell then repairs this break and researchers can either disable the target gene or insert a new sequence using a template DNA. This simple yet powerful process lets scientists edit the genomes of living organisms with unprecedented precision. So meet Emmanuel Charpentier and Jennifer A. Doudna for winning the Nobel Prize in Chemistry in 2020 for the development of CRISPR-Cas9 method of gene editing. And this is the summarized method. Moving on to the real-life example where CRISPR-Cas9 is used is sickle cell anemia. Sickle cell anemia is caused by one wrong letter in DNA. Gene editing can correct that mistake permanently. How is gene editing in cancer done? Cancer happens when cells stop following rules. Gene editing can switch off cancer-causing genes and boost immune cells to fight tumors, where immune cells power up and cancer cells are seen to shrink. Gene editing isn't just about humans. Crops can be made disease resistant, drought tolerant, and more nutritious. Next comes precision genomics, the next level of medicine. Here is the problem with traditional medicine. Same disease, same drug, same dose for everyone. One size cannot fit everyone, right? So, precision genomics says your DNA decides your treatment, different DNA profiles, different medicines. In cancer, doctors now analyze tumor DNA before choosing a drug. Right drug, right patient, right time. Tumor DNA is scanned and targeted with the drugs in a missile style. Now comes some ethical questions. Powerful tech brings big questions like designer babies, editing embryos, playing God. In the future, gene editing could cure genetic diseases, detect cancer early, personalize all medicine, and gene editing doesn't just treat disease, it edits life at its source. If this blew your mind, hit subscribe because biology is no longer boring, it's powerful. Thank you.